All right, so today's presentation is about what's in our creek, and it's really a biodiversity assessment of Enoggera Creek and Fish Creek. And before we start, we really want to acknowledge our partner for this study, SOWN, Save Our Waterways Now. They have been essential securing funding and the support. A lot of their members are also part of the Platypus Group here at the Gap Sustainability Initiative. And all together, we keep working toward achieving, hopefully, a reintroduction of the platypus in a few years' time in the Gap. So thank you very much to SOWN for their support. So what this survey is, it's called an eDNA survey. So what is eDNA? So eDNA means environmental DNA. It's all the genetic materials that we can find in one spot in an specific environment. So like yourself, you might lose some hair or a bit of skin that contain genetic materials. Animals and plants do the same in their environment. So this genetic material can be skin, hair, scale, feathers, could be gametes, eggs, mucus, saliva, fecal matter, urine, or even a whole organism when these ones are very tiny. So, so exactly what we've done, we sampled the water of the creek, and we've been looking at what genetic material we can find in the creek. So the advantage of eDNA is that it's a non-invasive method. We don't need to get the animals out of the water, so we don't disturb anyone. We just sample a little bit of water, and that's it. So non-invasive. It's very easy to do. Anyone in the community can actually sample the water. We will show you in a moment how it's done, but very simple, accessible to everyone. And it's fast to do. The advantage is where we can identify thousands of species at a time. So it's a really, really powerful tool. It also comes with some limitations. What we really want to highlight is it's important to understand these limitations when we look at the results of the study. So limitation of eDNA surveys is they are a snapshot in time and in space of the environment. That means that where we are taking the sample, it's what's happening at this exact location and at the time of sampling. So if we are not detecting an animal, could be that this animal was in the creek, but not at this particular spot, or not at this particular time point in time. The detection sensitivity. So not all animals or plants are detected the same way. Some are more easily detected than others. And also the eDNA degrades in the environment. And depending on the type of genetic material that is present, some might deteriorate faster in the environment than others. And it also depends on the reference database that is available. So what I mean is that all the genetic material that is analyzed by the company, they are comparing it with a reference database. Whatever is in the database, we can detect in the sample. But not all animals and plants are currently in a database. Now, this specific company, which is called Lab, it's in New Zealand, they are using the reference database GenBank, which is the most commonly used database worldwide. And as of October 2024, they had on their reference database over 580,000 species in there. So, we are detecting a lot with that. Anything you wanted to add? The methods, so very easy. So this is the typical kit that we are getting from the Lab company. It comes with one syringe, so each kit, one syringe of 50 ml, a little filter, that's the critical part, a little syringe with preservative in it, and a pair of gloves. And for each sample we collect, we use one of those kits. How we do that is we take the 50 ml syringe and we have to collect 20 times per site, per sample, water. because we need to filter a liter of water per sample. So that's simple math, 50 mils times 20 is 1 liter. And we have to filter it through this little piece here that contains the filter. Once we have passed through 1 liter of water in the little filter, we inject the preservative in it, and that's it. This little bit is sent to the lab, just a little filter. So with Andrew, on one day in October, we spent 7 hours together sampling the creek. We've done 3 samples per site, so it takes roughly an hour per site to do 3 samples, and we sampled 7 sites, which Andrew is going to show now. So we have one site at the dam, one site at Enoggera Street, Riawena Street, Witanga Park, Glenella Street, Walton Bridge. This little location pin should be actually here. I might have made a mistake when I entered the coordinates, so I will have to fix that. And then one in Taylor Range. So this one at Walton Bridge is when you have Fish Creek and Enoggera Creek connecting. So after the 2 creeks connects to one another. And then, as I said, we send these little samples to New Zealand to wider lab. And the good thing to know is that all the data that we are presenting tonight is publicly available. Any of you can go on the wider lab website and access the data. So today is just a summary of what we found, but yeah, feel free if you want, if you're interested, to dig deeper in some of the analysis. Andrew will show you how it looks like. It's a lot of data, and we'll tell you a little bit more about what we found.
Yeah, so when we get the data, it comes in an Excel spreadsheet, and it looks like this. So there's lots of information in there. Basically, first thing is, if a sequence of DNA that we've detected in the eDNA matches to a species that we know, we might get the information here like we have here, species name. Sometimes that can't be that accurate. We don't have sequences from every animal or plant, as Fanny suggested. So we might get a higher taxonomy. So it might be the genus or the family that particular DNA is coming from. So throughout this, it's a bit of a challenge when we're looking at the data to go, well, what can we actually read from this? The other key point that I want to make here, apart from the names, is also looking at the results that we actually have here. So we've got each of the different sites, and then we have these numbers under here, which refer to the number of reads or the number of different fragments of that same DNA that we actually detected. The thing with them though is you might have a high number because maybe there was a dead fish right next to where we were sampling, so it looks like we've got thousands of reads for a particular fish. Or it could be because there's 1,000 fish. We don't really know, so we can't directly take from this an abundance just because there's lots of reads. But what we can look at is the number of sites that we've detected a particular species or a genera of animal as well. And from that we can start to go, well, this looks like it's common in the creek because it's found in many sites, or it's only found in a few sites. Maybe something's going on there. So throughout the presentation you'll see I've referred to the number of sites in there. That's for your information to think about what that might mean, and I'll come back to that in a few particular points. And this is broadly looking at proportional, what the different groups of life or organisms that we detected in the eDNA. So the biggest group was bacteria. So 25% of all the reads corresponded to bacteria. I'm not going to tell you more about bacteria than that because I've got no idea what it means that we have all these different types of bacteria. Don't know how that works actually in the ecosystem of the creek. Followed by that, then we had the plants. So lots of plants of lots of different groups. Then we had the insects, which is a really good sign. Insects are platypus food, so we like to see that. the silates, which are a worm sort of looking thing. We've got other, which is really a data error. It shouldn't be in there, but that was a real mix of outliers. Algae, followed by fish, worms, fungi, green algae, and a whole heap of other different groups then in smaller and smaller numbers. So I'm going to start with the plants, and at the high level, what we saw was that we have subtropical rainforest. I saw that we've got some of the SOWN, the GAP rainforest books in here, and I was having a look at this, and a lot of the species that we've detected in eDNA are in there. So I think that it's really the work that we've done reinforces that those species are turning up. They're turning up in eDNA as well as in the on-the-ground surveys that have been done by SOWN as well.
If I can add something, sorry Andrew. It's very important to keep in mind that obviously these species, they don't grow in the water. We are detecting them because a leaf has fallen or pollen has come in the water, but it's just to tell us that this is in the environment of the creek. And obviously that's a wonderful work that SOWN is doing, keeping our rainforests alive and prospering. So that's a good sign that we are detecting it, but it's not growing inside the water.
Yeah. What I will say that we are missing is we don't seem to have aquatic plants turning up in the eDNA surveys. So not really sure what that means, whether there's not plants there, whether the sequences for those plants haven't gone into the library so we can't actually detect them. But at the moment, we still need to do a bit more analysis, and maybe there's some higher taxonomies that we can actually identify in there, but we're not seeing much, nothing's actually jumping out. And none of your classic aquarium plants either that you might just see that someone might have released. What we do have is a really broad range of different types of plants that we are seeing. When we looked across the sites, at every single site we sampled at, we had eucalyptus, so gum trees, and casuarinas as well. So they're the she oaks that you'll often see all along as well. But after that, again, this one's sort of proportional, showing you how many, it's proportional to the number of sites that each group of plants was detected in. And the key point here is there was lots and lots of different plants that we were detecting. But it's not all good news. When I went back and looked at the sequences that identified to a species level, 64% of those are either weeds or introduced species that aren't native to here. So we do still have a lot and a large diversity of weeds and introduced species here. Some of them will know, if you walk down the creek, you'll see Singapore daisy everywhere, but say West African tulip, it's a really bad one for our insects. When they actually feed on it, it actually kills them. And unfortunately, they're still around. I don't know if there's any right near the creek, but you still see them in people's yards around the gap as well. So these weeds are still a problem. And we had a couple of odd detections, and not really sure what to make of this. So we detected these two species, the trumpet thorn. I'm not going to try and pronounce the scientific name. And then this other one, Odontognia lutea, at Rowena Street. There's no records of these species in Australia. So my first instinct was, this is a bit of a mystery. We've got a fragment of DNA that's trying to match to a library that's not complete, and it's gone, this looks like what it is. So that certainly could be the case, and it's most likely what the case is. The one thing that sort of was a bit of an alarm for me is when I looked at these plants, they've got pretty flowers. And so many of our weeds have come in because they're pretty, they're in someone's yard. So these really just flagged to me of going, I think I'll keep an eye out and look for perhaps flowers that look like this around the gap if they are here, either in someone's yard or turning up in the creek already. Saying all that, it's most likely that it's just an error in the data. But we'll see what comes. We'll dig in now into the invertebrates. So this is things like your insects, your crustaceans, worms, spiders. So we had a good diversity of invertebrates. This graph here is actually roughly in the proportion of the number of each of the different groups that we actually found. So most of the invertebrates were insects, followed by worms, crustaceans, and lots of other things after that. So we had 68 genera, 115 species that we actually found. And glad to say there's some platypus food in amongst there. So really good to see that we've got prawns, we've got crayfish, we've got yabbies. These are your aquatic crustaceans that are in under the water, definitely good platypus food. We've got the dragonfly there. Justine might know more, but I don't think they would eat dragonflies that often, but the earlier life stages of dragonflies are aquatic. So they live in there. Platypus will definitely eat them as well. And then we've got the mayflies and caddisflies as well. So the mayflies and caddisflies, lots of different species found throughout the creek. But when we look at the freshwater prawns, the crayfish and yabbies, as well as then the dragonfly, it's only one or a couple of sites that we actually detected those in. So this is another one where we're just like, what does this mean? We could have just missed them. Maybe there's something about their eDNA doesn't turn up as easily as other species. Or it could actually mean that perhaps the abundance of these animals isn't very high in the creek. They're there, they're surviving, but they're not thriving. So this is one that we've really taken away of going, we want to learn more and understand more about what this looks like around the rest of the creek. And we also have introduced species. So yellow crazy ants. I think anyone who has done bush care along the creek will know that they're everywhere here, despite the best efforts of lots of levels of government to try and do something about them. They're here, they're continuing to grow and spread as well. And then we had a number of introduced snails. I don't know if anyone knows what the impact of these snails are in the creek is, how they've actually got here, presumably through an aquarium trade. They could also just be hitchhiking and accidental introductions through, boats, anything that's gone into the water that kept them alive as well. But we found a few different types of snails turning up as well. We don't have it here, but we also got told that there was one reed, so one sequence of fire ants turning way up at Urala Street as well. And I think that's a really good example of, anywhere else, and you might go, who knows if that's real or not, but we do know that fire ants are in the area, so it is quite likely that there was something there that's washed in, telling us that, fire ants are in that area. Now we'll get into the cooler animals with a backbone. So into the fish. So we still had some good diversity of fish. We had 9 genera, 19 species of fish that we actually found in the creek. And there were lots of native fish, which is really positive. So in all the sites we were detecting longfinned eel, which I'm not sure, they are often seen in the creek, so it's really good that they're turning up in the eDNA as well. The gudgeon, which are sort of a small fish, but they're really colourful fish, often well known. If you go down to Walkabout Creek, they've got a really nice display in there of them. But we detected striped gudgeon, carp gudgeon, purple spotted gudgeon, which I know Leo, who presented to us on fish, was really excited about seeing that one as well. Empire gudgeon and firetail gudgeon. I think the firetail gudgeons were just up in the dam, but they're around. So it's good to see a diversity of these small fish turning up as well. When we look at the rainbow fish, so they're a bit bigger than the gudgeon. There's just the one species that was turning up, the crimson spotted rainbow fish. But this is, my understanding, the native rainbow fish for our area as well. So it's really great that that's turned up. We're also seeing Australian bass. They've probably come in from, because the dam is stocked with Australian bass, so I assume these are survivors down here. Certainly there's a weir that moves, so Australian bass, they go from the freshwater down to the saltwater to breed, and then they swim back upstream as well. But we know in Enoggera Creek there is a weir that actually blocks their movement unless it was actually in flood as well. So that's sort of why we think it probably is more of a dam escapee that's there than one that's naturally bred and come up from, say, the Brisbane River. But we did also detect pest fish. So mosquito fish, Gambusia, they're everywhere in Australia. Not surprised at all that they're here, but they are a pest fish and they harass other fish as well. So that was there. And then a green swordtail, which I'm sure is an aquarium escapee, but they would be breeding populations of those turning up in the creek as well. When we look into the reptiles and amphibians, this is all that we detected that's native. So we have the eastern stony creek frog. That was just turned up at Walton Bridge. This is native to the area. It's good to see that it's still here. Although it's not endangered, it has been assessed, which suggests that there are concerns for it. And we know that nationally their populations are decreasing. So it's still something to keep an eye on as we go. We also have the tusked frog. We know that they're in the creek. We hear them all the time when we're down there doing bush care as well. Great that they turned up in the eDNA and we know that they're there as well. Same situation with the tusked frog though. Although it's not endangered, it has been assessed. So there is concern from it, and we know that the populations are decreasing of tusked frog as well. So again, another one for us to keep our eye on and to continue monitoring as we go along with this project. And then we've got two species of turtles that turned up. Eastern soft-shell turtles and eastern short-necked turtles. Does anyone know about turtles here? Because I know nothing about them. I think we can assume that they're probably native then. It looked like from their distribution, they're well distributed across the east coast.
There used to be a lot more of them before the last big flood.
Okay.
You'd see them fairly regularly in that stretch between Ryawena Street down to the Allowa Street Bridge.
Yep.
But yeah, don't see as many anymore. I think they're all washed away.
Again, good that they're here, good that we're detecting them. And I think no one would be surprised that we have cane toads as well. And they were detected everywhere in every site that we took samples from. Cane toad eDNA was detected there as well. So not sure that there's much that we can do about that. I think they're going to be here for a long time yet. So birds are an interesting one because a lot of birds aren't aquatic. So how their eDNA would be more restricted in how it's getting into the water itself. But we were detecting native birds, dusky moorhens, bush hens, your noisy miners, as well as your pied cormorants as well. This was the only birds that we did detect though. There wasn't more there that we don't know about. And a number of invasive birds. So mallards, I didn't realise that mallards were not native until I did this. And now I know that mallard ducks are not native to Australia and they turned up as well. Common starlings as well, which I've never seen around here, but others might have. Their eDNA was turning up. Common miners, which unfortunately are becoming much more common in the Gap as well. And then spotted dove as well, which you do see around as well. I will just make a point here is that not every introduced animal or plant is a problem. So there's very much different categories where you might have some introduced non-native plants in your garden that are completely fine, it's not going to spread, it can't breed, and it only exists because there is a nursery industry. Whereas you do have other species, like the common miner, that are invasive, and they cause problems, they chase other birds out and affect our native wildlife as well.
Back in the day, I thought the common miner was called the Indian miner. Or is that a different species?
It's exactly the same species.
Yeah. So the noisy
probably
miner
got a new common name because it's more politically correct.
So noisy miner is the Australian bird.
Yes.
Common miner is.
That's right. So that's the brown one with that yellow beak. Versus,
Oh.
can you go back a slide?
And a different spelling.
Ah.
Yeah, different smelling, and that's the grey one with the yellow beak. So the sort of, once you see them, you'll see them and you'll always pick them up. They both move around in flocks, and they're sort of, yeah, living groups. And then the mammals. Again, spend any time down the creek, you're probably not surprised to see grey flying foxes down there as well. I do know that we have a few other flying foxes that come in to the creek at different times of year as well. They're not necessarily always there, but we are detecting them. They are a vulnerable species. And in fact, with climate change coming, they're in real trouble because they're very sensitive to heat as well. And a few degrees change and their populations are going to get hammered. And we'll have to see how they go. And then we also detected common brush-tailed possums as well. No ringtails, no, what was the one that we saw in SOWN the other day?
Short-eared. Short-eared.
Short-eared.
Yeah.
They didn't turn up either, but that would have been good.
So the ringies are dying out.
They're around.
They're around still, they're definitely around. And then introduced Louis down by the creek. We definitely have domestic dogs, DNA turning up in the creek as well.
But not cats.
Not cats, not foxes.
The cats and foxes are regular offenders on trail cameras, so they are definitely there,
They're not seeing.
but they're just not getting into the eDNA. Yeah.
They're not swimming much.
Yeah.
So there's lots of things that we've got from the eDNA work that we don't know what they are, how they fit in the ecology of the creek, what they mean, something like that. And some of those that turned up that we don't know what it means, but they're there. So freshwater jellyfish, I know some of the people I've talked to didn't even know that freshwater jellyfish existed. But we detected this species here, which appears to actually have a global distribution. It turns up in lots of different places. Makes me wonder whether actually the taxonomy, they're actually different species, but there's not enough freshwater jellyfish experts in the world to pull everything apart. But we definitely have them there. We also have these hydras, which are closely related to a jellyfish as well. And we had a few different species and groups of those actually turning up as well. Although that's sort of a fancy photo there, they are of a size that if you know what you're looking for, you can actually see them down in the creek as well. And then freshwater sponges turned up as well. So sponges are one of those groups of animals that attracts really strange people to study them. So if you've ever met a sponge expert, they'll talk to you all day about how amazing sponges are. But the one fact I always take from it is that sponges, they don't have a skeleton like we do. They actually have a silica skeleton. So it's actually like glass. So you can actually see these amazing structures left over once the rest of the flesh essentially has gone of glass left over from sponges as well. But we have a few different types of sponges turning up as well. So amazing diversity that we have. So what didn't we find in the creek? We're not really surprised, but we're always hopeful. Platypus. We did not find any platypus DNA in the creek at all.
And that's important to mention that this wild lab, they have a marker specific for platypus, so their lab is very sensitive to finding platypus. So
Yeah.
in that case, in the case of the platypus, we don't find it, it's very likely it's not there.
And this really reinforces previous eDNA work that's been down in the creek, as well as all of the anecdotal evidence that they are extinct from Enoggera Creek. What we were surprised at was sort of hopeful that we might detect Ricali. We have had some reports of people seeing them more recently, which is really positive. And if Ricali can survive, they're a good sort of indicator that maybe platypus can survive as well. So we're a bit disappointed about that, but Ricali, they can move. So maybe they just upped and moved somewhere for the moment, or maybe they're just a bit more downstream, bigger pools, something's going on. Lungfish. So we have heard reports of lungfish turning up in the system. So we're sort of interested to see whether that turned up at all. But we didn't have any eDNA from that published as well. So again, they could be there. Maybe there's something with their DNA that it's not detected well. But we didn't find it this time.
In my experience, you tend to get the lungfish if the dam spills. Okay. And I saw one about 3 weeks ago, just down the bottom school road, about 40 centimetres long.
Okay.
But it was really struggling to survive, and I would say it was probably dead within the next. But they're there, especially after the dam spills.
I will add lungfish. There is arguments about what their actual native range is. From my understanding, currently they do think they are more traditionally restricted to the Mary River catchment basin, but we are just downstream from that, and they are here now. They're probably being moved here, but it's hard to always know what the true history is. And I also believe in fossil records they were found much more commonly along the east coast as well. So I think if we saw them, that would have been good. It's not a problem here. It's not something that's dramatically affecting the ecology or having negative effects.
There are records of lungfish being released into Enoggera Dam in the 1930s, as they did to several dams around, hoping this would broaden the population.
There you go.
They're at Carlo in the Brisbane River
Yes.
Yeah.
because I've caught them.
Yep.
Ugly.
I've done the same.
You tried to check me.
Yeah, after the last floods in New Alice Street Gardens, they had one in the banana patch. That when the water went down, they found, and they had to bury it.
So hearing this sounds like they're probably definitely happy in the dam. One thing that I was really surprised at, but really encouraged at, is tilapia. We did not detect tilapia anywhere, and I really expected that they would just be here. I know they're all through the upper stretches of the Brisbane River otherwise, and obviously Enoggera Creek is connected to Brisbane River, so I just assumed, yeah, they'll be there. But they weren't. So that's really good. Tilapia, they can really affect the bottom of the creek. They'll go in, they'll completely change what's in there and what the creek looks like, destroy a lot of the native plant life. So it's really good sign that they're not there. Does anyone know if tilapia are definitely in the dam?
Seen them in Enoggera Creek.
You have seen them in the
Yeah,
Enoggera
just
Creek?
by the doggerfish area of New Alice Street.
Okay. And also wasn't found. So again, back to the Gap Rainforest Statement of Significance, there's 3 species in there that were identified that were vulnerable or critically endangered species. So Macadamia integrifolia, so one of the species of macadamia, we didn't detect it. We do know that there's a number of those trees along the creek in Enoggera though, and we can see them. So that's okay, there's other ways to detect that. Then we're looking here at the, my mind is failing me of their common names. The myrtles. Both of these species have been severely affected by myrtle rust, which turned up here in Brisbane about 10 years ago. And these are the species that we're concerned will go extinct eventually. I know that some populations of these have been translocated west of the Grudge Voting Range in the hope that myrtle rust wouldn't get there. Saying all that, myrtle rust has gone south to Tasmania, west to Western Australia now as well. So we'll have to go and see. I know last year that I saw one of these down near Walton Bridge. So we know that there are some physical samples. I haven't been there in a while to see if that tree or population is still surviving or not as well. But we didn't detect them through any of the work. So I think the big question that we sort of came out of this was just really going, there's lots of stuff that we've detected, which is really positive. We have some level of biodiversity, but how do we now take this data to sort of assess, is our creek healthy? Is our creek suitable for platypus? So I'll hand over to Fanny to talk a little bit more about some of the indications we've had and how this data plays in with the other work we're doing with the creek monitoring.
So there are several ways to look at indicators of the health of the creek. One that WildLab is providing is a Taxon Independent Community Index. That's an index they created themselves using the data from eDNA surveys. So now this indicator has been made for New Zealand rivers. It's on the test for Australian rivers, but my understanding is New Zealand rivers and Australian rivers are not that different, and they believe that the results should be very similar to what we found here. So now this indicator, this index is not public. So if you go on a website, WildLab website, you look at the data, you will not see that index. However, WildLab did send us the results in their data, like the data set that we have. So how it works is we have different critters in the rivers. Some animals will be very tolerant to water conditions. If there's pollution, they don't really care, they will still be there. Others will be very sensitive to the water quality and the condition of the creeks. And they assign a number to each of those different animals depending on their sensitivity. So a low number if the species is very tolerant, and a high number if the species is very sensitive. So what we found at all sites is using this indicator is that the score was below 90, and here you have the scale, and below 90 corresponds to a poor health condition of the creek. All the sites were poor, or even one was very poor. So not completely uncommon for urban areas. So if you go in places that are more isolated and pristine, you would probably have a better index. But that's one indication using that. Again, made for New Zealand rivers, so to be taken with caution for Australia, but it's likely that this is adaptable for Australia. Another way that we can measure the health of the creek is by doing a macroinvertebrate survey. So that's going into the creek, getting some water samples, and then manually, so you're really invasively taking out the animals from the creek, sorting them, and identifying them, counting them. So that's what we've done already twice at Enoggera Creek. So we've done one in October, that was our training actually. We've been trained to do the surveys by the previous BCC catchment officer, Hannah. And then we've done another one on the 25th of January, again for a group to train and be confident with the surveys. And this type of survey, that's what we will be doing Sunday with the community, and you're all welcome to come and help us do this. So by sorting all the animals, so we have a signal score that's indicative, again, same as the previous slides. We give a score to each animals. Some are very tolerant to water conditions, some are very sensitive. So with that survey, we do exactly the same thing. And we count how many there are of each. So that signal score is the number of sensitivity, and also take into consideration how many of those animals we had. And then there's also the number of types of bugs that we detected. So to make it short, all our two surveys, they had a result here. We had a signal score below 5.5, and we had a number of types of bugs between 0 and 10. And here, it's indicative of pollution. So again, not great indication of the health of the creek. So now using two different index and two different ways, we're getting to the same results, but yeah, could be better. Now, there's a lot of things we still don't know. This is just the start. We don't know how these results, for example, compare to other creeks nearby that have platypus populations. We don't know how this data evolve after a big flooding event, for example. Again, what we presented today is only a snapshot of one day in October when we sampled the creek. So what happens after a big flooding event? What happens at different seasons? We don't know. Are there contaminants in the creek? What kind of pollutants are there? Are they affecting the health of the creek? The big unknown of the aquatic plants, is it we didn't detect them? Is it they are not there? Do we need them? So that's something we're hoping to maybe find an expert to help us look again at the data. Again, Andrew and I are not experts in that field, so might be worth having an expert looking at the results and see if this is something we haven't found. Why are there so few frogs, reptiles, or even native birds? And what other species could be there that we are missing? And then the big question is, why did platypus go extinct in the first place?
Just something I want to add to that. I know we've presented poor results, and that's not good. That's really telling us we want to do a few more surveys, but if we keep getting these results, we know something is going on and we need to do something to try and improve it. The good news for platypus is that they're not super fussy. When they've done platypus surveys, even around Brisbane, you're finding them in drains behind industrial estates and creeks in lots of urban areas where they are surviving. They're not necessarily thriving. We know locally their populations are decreasing, so there's definitely problems. But there is a glimmer of hope there that this is one species that could survive, and if we put it back in, maybe there is a good chance, particularly if we can improve some of these indicators of creek health. It doesn't have to be perfect. We don't have to take it back to, pre-colonial Australia. We just need to get it good enough that they can survive and hopefully thrive where we are.
So where to next? Well, we need a lot more data. This is only the start. We started with no data. We didn't know at all what was in the creek. So that's the first step, but data is going to be crucial. Why do we need data? Because anything that we need to put in place to better the creek will need proof. We will need to be able to show the council, SEQ Water, government, this is what we have, this is what we need to do. And that all comes down to data. Without data, we cannot do anything. We cannot prove anything. So we need to continue to collect data. So that starts with our monthly creek monitoring surveys that we started and that we will open to the community as of Sunday. We are hoping to do yearly eDNA surveys at the minimum. That would depend on funding. We need nearly $7,000 to do the eDNA survey that we've done. Why do we want to do more eDNA analysis? Well, that was only one snapshot. We need to repeat. Do we find the same thing? And also to see the evolution over time. If we are implementing measures to improve the creek, we want to see if those measures are working. If we are not doing anything, we need to keep monitoring, is the creek stable, is it getting worse, just know what is happening. We are also hoping to start analyzing the pollutants that are present in the creek. So Monica has a contact at UQ and we are hoping to involve the students on this. The next step, compare these results to neighboring creeks that have platypus populations. And we're very lucky to have Justin in the room at the back. Justin is a PhD student with Dr. Gilad Bino, who, for those who were there in 2023, came here and presented the story behind the reintroduction of platypus in the Royal National Park in New South Wales. He was a researcher involved in all that process, so that was very interesting. And he's helping us as well, and he's supervising Justin. And so Justin is already using our data, and comparing with results and data that she's collected in the Albert River in Logan area, that has a platypus population, and also Mongill Creek, which is very close to us and has a platypus population. So we will welcome Justin toward the end of the year to present our data, and so that will be the next step in the story, and you will be able to learn is our creek at a similar level to the Albert River or Mongill Creek, or what's different, and so that would be very useful. As we said, like it would be continuing collecting data to identify, okay, what's the problem with the creek? Is it because we are missing these aquatic plants? Is it because there's pollutants? Is it because of some other reason? But before we can implement a plan, we need to understand the issues, and that all goes back to data. The next step is to do habitat assessment, which we haven't done yet. So for platypus to be reintroduced, they need water and they need food, but they also need a good place to live. That means water holes that are permanent all year round, that don't dry up even in summer. They need to be at the right depth, and they need to have suitable banks so they can breed their burrows to reproduce and have their young. So that's a very important assessment. We need to identify best permanent water holes. That's the water holes that we will want to keep monitoring over a few years' time to see if the habitat is suitable for reintroduction. This is also these water holes we will probably be monitoring and be in contact with SEQ water to make sure that the water level in these holes stay sufficient all year round. And obviously we need all of you. We need the community to be behind the project and contribute in raising awareness about our creek, about the issues we're currently facing, and about reintroducing platypus. We need to make sure that we don't continue polluting the creek, having shopping trolleys floating around. There's also the issue of dogs, that if we reach the stage of we having a healthy creek, it's an habitat that's suitable for platypus. We won't be able to introduce platypus if people continue to just let their dog roam around free, and we will have to address other pest species like foxes as well. But that's another step. So still a lot to go, but very exciting. It's just a start.